Hey biology students, this is Mr. Gales, and I think this is actually going to be the last screencast of the year. We're running out of days real quickly. Mr. Workman and I talked about getting you the information you'd require for the end of the evolution exam, so this is going to be it. I'm going to try to go through this relatively quickly. You're looking at pages 93 to 95 in your evolution packet, and what you should see is there are a series of clips. It says watch this clip, and then some questions that need to be answered. You can answer those questions primarily by watching the video, but also by kind of listening for what I'm going to discuss here. This information, what we're going to primarily touch on are comparative embryology, comparative biochemistry, and comparative anatomy, which are what we consider evidences for evolution from living things we can actually take a look at and see. So the first type of evidence comes to us from comparative embryology. And you should remember from earlier in the semester that embryos can be seen as organisms that would be at an early stage of development. A blastocyst, for instance, is an embryo that's made up of only a few hundred cells. Now, comparative embryology is valuable for us because we can see similarities between organisms at similar stages of embryonic development. I'm going to start off with a video clip here, which is called Common Pasts, Different Paths. And what you should be listening to during this video as we play this is kind of, the questions that are listed on page 93. When do human limbs first become visible and what do they look like? And how do the embryos of humans, pigs, and birds compare at this early stage of evolution? What I want you to be thinking about is why do you think the embryos look so similar, and what features would all embryos of vertebrates share during their development?
Human limbs begin as buds, seen as early as 5 weeks in this embryo. At this stage, an arm bud, seen here, appears very similar to this leg bud. By 10 days, the limb buds are shaped like tiny paddles, and distinctions between arm and leg buds are visible. The distinctions can seem slight. A human arm at this stage still looks like a pig's embryonic front leg. Bird embryos at this stage show extraordinary similarities to those of humans. The left bud, looking so much like a human arm, will ultimately develop into a wing. Each species starts with a shared beginning, but takes a distinct course, acquiring distinguishing traits of its own.
All right, so what we see here when we look at the embryos from the pig, the human, the chicken, and the fish, we see that there are similar structures that are found throughout the development, features that would be common to all vertebrate embryos. And we're going to go through kind of a list of those here and think about why these embryos share these characteristics. When we look at vertebrate embryos are embryos that have a backbone, right? Organisms with backbones or vertebrates. We see that all these embryos share some common features, and one is an eye spot. We saw very early in the development the eye, which is just a patch of or a collection of photosensitive cells, and obviously that becomes more complex as the organism develops. You can see here on these pictures of the various organisms, you can see the eye patch here in each of these. Gill pouches. Gill pouches are structures that are left over from an earlier ancestor that are no longer necessary in organisms that breathe air, but for instance in fish they develop into full gills. In organisms like animals, for instance, like chickens or pigs or even humans, those gill pouches are sort of the anatomical features of where the gills would develop. They don't really develop into gills. You can't really breathe with gills even in the womb, but those are the pouches where the gills would have formed. And then there's something called a notochord, which is the essentially the area where the spinal cord will develop, and we see that here in each of these types of embryos. Now, the big idea, this is the key idea here, comparative embryology sees similarities between diverse embryos because all of these embryos share genetic ancestry. They share a common ancestor. And that's going to be a very common theme as we go through this presentation. The common ancestor of all of these organisms had these characteristics as well, and that's why we continue to see them in our embryos. Even though as we develop, we lose some of those characteristics that are present in us. Now the next video is called the Genetic Toolkit. I'm going to open that one up, and this is going to be number 2 on page 93. And what we're really going to listen for here, I want you to be paying attention to, again, the idea of why are there so many similarities between living things. But we're going to begin by looking at what does evolution tinker with that leads to variation in living things, and then what happens when the gene for the development of mice is placed into fruit flies. Why is evolution simpler than we thought, and why do animals resemble each other? So let's take a look at this video here.
So what do we learn by looking at 600 million years of animal history? Evolution's tinkering with mammalness to make whales. In the same way, it's tinkering with fishiness to make tetrapods. And it's tinkering with animalness to make all the different body plans that we see. All these different creatures are variations of the same theme, restated over and over again. The question was, what was evolution tinkering with? One of the remarkable discoveries of the last 20 years is that evolution's not tinkering with the bodies. It's tinkering with the recipe, the machinery that builds bodies. What is that recipe? What is that machinery? It's the genes.
Scientists had long suspected that embryos held clues to how animals evolve. All embryos start out as clusters of nearly identical cells. But soon, an embryo partitions itself into specialized segments, which develop into the final form of the animal. What controlled this process? How did the embryos know what shape to take? One researcher, Dr. Ed Lewis of Caltech, studied this question for 30 years by crossbreeding thousands of flies. Lewis's work led him to a controversial idea. He proposed that a surprisingly simple mechanism was shaping embryos. He wrote that each segment of the fly was being directed to grow by a single gene. A small set of genes, a kind of genetic toolkit, appeared to be laying out the entire body. In 1994, Walter Gehring of the University of Basel isolated the gene that triggered the growth of eyes in fruit flies. The gene was called eyeless, because flies without it developed with no eyes. Gehring knew of a gene in mice that worked in the same way. He wondered, were the two genes the same?
And this question we tested by taking the mouse gene and putting it into fruit flies to see whether flies can understand the message of the mouse.
Gehring replaced a fly's gene for eyes with the mouse gene.
And to everybody's surprise, the mouse gene works perfectly well and can induce a compound eye in the fruit fly.
The fruit fly grew normal fruit fly eyes using a gene from a mouse. Not only did the two creatures use the same mechanism, they used the same gene.
So what this means is in some ways, some sense, evolution is a simpler process than we first thought. You think about all of the diversity of forms out there. We first believed that this would involve all sorts of novel creations, starting from scratch, again and again and again. We now understand that no, that evolution works with packets of information and uses them in new and different ways and new and different combinations. without necessarily having to invent anything fundamentally new, but new combinations.
Suddenly, the commonality of form among animals was understood. Animals resembled each other because they all used the same set of genes to build their bodies. A set of genes inherited from a common ancestor that lived long ago.
And there's only one inescapable conclusion you can draw from that, which is if all of these branches have these genes, then you have to go to the base of that, which is the last common ancestor of all animals, and you deduce it must have had these genes. So the whole radiation of animals, the whole spring of animal diversity has been fed by essentially the same set of genes.
So what do we learn?
When we talk about the evolutionary relationships between lots of different kinds of organisms, big idea that needs to come through here is the genetic similarity, the genetic ancestry that all of these organisms contain. So when you look at all the different kinds of animals, all the different diversity of animals on our planet, the reason that they have so many similarities is because they are the product of evolution, and they all were produced through this process from a common ancestor. All right, we're going to move on to the next subject here, which is comparative biochemistry. Comparative biochemistry looks obviously at the biochemical similarities between living things. And we know that all life is based on organic chemistry, which are carbon-based compounds, and that they all use the same molecule as a blueprint, which is DNA. That's the informational molecule. All living things undertake similar chemical processes. Like, for instance, bacteria, algae, and plants all do photosynthesis. And finally, similar organisms have similar genetic code. And we've talked about this in class before, but we know that humans and our closest living relatives, the chimpanzees, share nearly identical genes, 94.4% identical gene sequences. So let's take a look at this common genetic code. The first video that we're going to take a look at is going to be, I believe it'd be page 94 in your packet. It's called the Common Genetic Code. And you're going to look specifically for some information about the mutant yeast cells and how they differ from their normal counterparts. And then what happens when the gene for humans, the controlling cell division, is placed into the mutant yeast cells.
Our DNA, the genetic blueprint that makes each of us who we are, also unites us with all of life. Scientist Paul Nurse knew that humans shared fundamental genetic instructions with other living creatures. To test if these instructions could be understood and acted upon by other organisms, Nurse chose common baker's yeast and designed an experiment to see if it could decode and follow the instructions in human DNA. Yeast is single-celled, but like human cells, it needs to grow and divide in order to survive.
What you can see here are growing normal yeast cells. Several days ago we spread these yeast cells on this plate, and they've since then, each single cell has divided many times until it forms this visible colony, as we call them, on this plate. And I'll just put this here because we need a much higher magnification. So if I look down here and move the petri dish around until I can see something growing, these individual cells are like short sausages. They continue to grow, and then when they reach a certain size, they divide from one cell into two. But we can get mutants which don't behave this way. I've got some on this plate over here, and they're growing much less well. And if we look now under the microscope with these, then they look rather different. I'll just try and find some for you. They're like very long, elongated sausages here because they cannot divide.
Nurse used a simple method. He gave the mutant yeast cells human DNA taken from a library of human genes and watched to see if they could repair themselves.
So now we just have the pellet of yeast at the bottom, and I want to resuspend it in this liquid here. What I'm going to do is take a little bit of this gene library out of this test tube, and then add it to the yeast cells. Whirly mix it to mix it all up together again. And in there, we've got these millions and millions of yeast cells surrounded by these human genes. And each cell is going to take up one or two of these genes. We have to sterilize this glass rod here. And then spread it all over this jelly dish here. If there is a human gene in this library that can perform the same job as the gene that is defective in this mutant strain, then this mutant strain, which takes up that human gene, will now be able to grow and divide.
The last common ancestor shared by yeast and humans lived many millions of years ago. Since then, they have taken quite separate evolutionary paths. Would the yeast in this experiment still recognize the human instructions well enough to follow them? The yeast cells have now had access to the human genes for 72 hours.
I can see that those cells that have got the human gene are dividing quite normally, but those cells which have lost it have become highly elongated and stopped division. So this has really worked. This gene now is actually really allowing these defective cells to grow and divide.
I think it's rather touching that we should share genes with a worm. I mean, in these days when we think of the environment and we think of other living things, the fact that we share a gene pool with all living organisms, including the worm, including the lowly yeast, doesn't that mean that we should have a greater respect for all living organisms? I don't feel, in fact, or any the less for that. I feel ennobled that we share this gene pool, and it makes us respect all living things, all living organisms.
All right, so what we saw was that when you have a mutant yeast that can't go through cell division, it gets short. It doesn't divide. It becomes elongated because it can't divide to keep its normal short length, and it's lacking a gene that controls the cell cycle essentially. So when we put a human gene that controls the cell division into that same mutant yeast cell, the cell cycle is resumed normally. And what this indicates to us again is that there are common genes not only that control the development of an embryo like we saw in the previous film, but here we see the similar genes in widely different organisms that control the exact same chemical process or cellular process inside the cell. It's fairly amazing when you think about the fact that the gene that controls the cell cycle in you and the gene that controls the cell cycle in a yeast is essentially the same. So again, 4 major biochemical similarities between all living things: organic chemistry, DNA, similar chemical processes like photosynthesis, cellular respiration, enzymatic reactions, and then of course similar organisms having similar genetic code, right? The more similar two organisms are, the more similar their genetic code will be. And we're going to take a look at an example here of where that comes into play with human evolution, and the video is called "Did Humans Evolve?" This is number 5, again on page 94, and we're looking at a couple questions here. We share 98% of our genes with chimpanzees. Why would you guess that our genes are so similar? And at what rate do primate genes change is what we want to look at.
Did humans evolve?
Certainly, that's one of the most exciting insights that Darwin and others gave us, is that humans are not separate from this grand scheme of relatedness of all life on planet Earth.
In all of our distinctiveness, it can be hard to understand that we are related to even the simplest forms of life.
No candy at all. What would you say?
For many people, it's even difficult to accept our close evolutionary relationship to other primates. Part of the problem in understanding our relationship to other living primates is to look at the difference between a relative and an ancestor. Look at your own relatives. Here's you and your biological mother and father. You are related to them and descended from them. Your mother's sister is your aunt, and her child your first cousin. You are related to them, but you are not descended from them. When you look back at generations before you, you can see your ancestors. You and your cousin share 2, very close in this case, common ancestors, your maternal grandmother and grandfather. If you keep looking back at your ancestors for about 5 million years, you will arrive at the common ancestor we share with today's apes.
We didn't evolve from chimpanzees, but the common ancestor to both of these had a skeleton that was pretty similar to what we see in a chimpanzee. It got around on all fours. But through the years, with evolutionary changes, this skeleton, the skeleton of a four-legged creature, was modified for a different kind of locomotion, a different way of getting around.
These differences may look great, but at the genetic level, the differences are minimal, as Dr. David Page's research demonstrates.
In the last few decades, we've seen a completely new kind of evidence emerge that demonstrates unequivocally the close relationship between humans and chimps. And that evidence has come from DNA. This is DNA. We've got DNA, chimps have got DNA, bacteria have got DNA, petunias have got DNA, crabs have got DNA. Every living animal, plant, fish, frog has got DNA. And if we compare the DNAs of any two species, we can establish how closely related they are one to another.
The sequence of the 4 chemical building blocks of DNA determines the traits of all living things. These are represented by A's, T's, C's, and G's. Each generation passes on this chemical text to its offspring. Occasional mistakes in copying, mutations, can result in new traits. Page takes part of a gene in humans and aligns it with the corresponding sequence of the same gene found in chimps to compare the letters.
And what we see is that these two sequences, the human and chimp sequences, are spectacularly similar. They are, in fact, 98% identical. They're just a couple of spelling changes. Why are there only a couple of spelling changes? Because we and chimps had a common ancestor only a few million years ago. If more time had passed since we had our last common ancestor, more spelling changes would have accumulated.
To make that point, Page shows the same gene sequence of a mammal that is very different from humans, the rat.
You see that there are far more differences, let's say, between the human and the rat than between the human and the chimp. That's because our common ancestor with the rat lived about 80 or 100 million years ago, and there's been much more time for spelling differences to accumulate. We've learned that in primates, DNA sequences are changing at a rate of about 1% every 3 million years. In other words, about 5 million years ago, the precursor of humans and chimps lived. And we can see that. We can, in a sense, read that right out of the DNA sequences. In the 19th century, in Darwin's time, it was audacious to claim that humans and chimps were closely related. There wasn't that much scientific evidence. But in the 20th century, we've seen the fossil record emerge.
In the 21st century, the combination of fossil record, physical features, and molecular evidence leaves no doubt that humans and apes shared a common ancestor several million years ago. And humans and apes, like those early primates, have continued to evolve ever since.
And like the students always say when we show this video in class, thank the flying spaghetti monster that we also evolved pants, because that was not a good look. All right, so humans and chimps share 98.4% of our genetic information. Why are our genes so similar? Well, it's obvious we have a very recent common ancestor. What we saw here was the more recent the common ancestor, the more alike the genetic material will be. If we extrapolate out, approximately 5 million years ago, the common ancestor of modern humans and modern chimpanzees lived. And so what we look at is the gene sequences to see the similarities there. All right, moving along to the next topic, we're looking at comparative anatomy. This is going to be question number 6 on page 94 in the packet, looking at vestigial organs. These are organs that are inherited but not used by modern organisms, so they would be present on the body or in the body, but they could be greatly reduced in their structure or obviously in their importance. And some examples here: hip bones in pythons, appendix in the human, and the tailbone in a human. And there's going to be a quick little video I'm going to show you here, which does a great job explaining vestigial structures. So we're going to jump out here and go to YouTube for a moment, and this is Hank, and he's going to talk to you about vestigial structures.
Unless you've had it removed, you, like every other person in the world, have an appendix, which is a kind of a stupid thing to have in your body. It doesn't do much. It kind of cuddles with your intestines. Don't imagine your intestines don't mind having someone to cuddle with. It does provide that function. It also holds on to some gut bacteria, which is nice. But it pretty much does nothing for your entire life until the day when suddenly it has an uprising and decides to kill you. The appendix, along with some other completely useless things in your body, like wisdom teeth and your tailbone, well, they did not escape the notice of one Mr. Charles Darwin. He was a scientist. You may have heard of him. So Darwin wrote this book. It was kind of important, called On the Origin of Species. And in that book, he said that these things were vestiges of bygone eras. Times when the ancestors of humans somewhere down the line had a use for a tail or some extra teeth for chewing meat or an organ to process a bunch of plant cellulose. Now, Darwin here was not the first person to notice that humans, as well as some other animals, are tricked out with various equipment that they don't need. Darwin suggested that animals evolve within a certain set of circumstances, and they develop body parts based on those circumstances. But when those circumstances go away, those body parts, they fall into disuse. But physical evidence will remain. And the amazing thing is that he was totally right. They're called vestigial structures. They're not always organs, so we say structures. And they're basically one of the many things about our bodies that leads us to believe that we evolved from other species. Here are some other examples of cool vestigial structures that we have. Goosebumps. Goosebumps are a reflex rather than an organ, but they are considered to be vestigial in humans. The pilomotor reflex is this thing that happens when a bird or mammal gets cold and wants to warm up, or when it sees a predator and it wants to warn that predator to step off. There's this tiny little muscle at the base of every single hair follicle that will contract and pull the hair erect. Just because I said erect, that's not funny. And for a bird or a dog, that might help it get all puffy and look bigger to a predator, hold on to more air so it can stay warm. The tailbone. Our tailbone, or the coccyx, is this little vestige of a tail, because our ancestors used to have tails, and I'm sure Stan won't mind if we show off his butt here. And there you have a little tail. Can you get a zoom in on it? Zoom in on it. Yeah. See? The little butt tail there? Got a little tail! And so do you. That was excellent work. You're a really good actor. We see tailbones in all great apes, and we even see them in the fossilized remains of our ancestors all the way back to the Miocene period, when our ancestors were first trying to grab onto things with their newly opposable thumbs. So now all we have is this tiny, itty-bitty stump of a tail. But it is not totally useless. That little bone is now the anchor to which all of the muscles that help us control our continence, if you know what I mean, are stuck to. So thanks, tailbone. That is an important job, and I appreciate it. And our final vestigial structure, the wisdom teeth. We humans have become such delicate flowers that our mouths no longer have room for all the teeth we need in order to eat all of that woolly mammoth meat that we don't eat anymore. So now that people are all agricultural and omnivorous, and we generally tend to cook our food well, our jaws are now too small to accommodate all of the teeth that grow into them, and that results in modern humans frequently getting impacted wisdom teeth. I actually have one of mine left, because they took out 3 and they couldn't get one. They left the hole, but also the tooth was still there, so I'm worried about this happening to me. But 35% of people born today do not have wisdom teeth at all. So we are actually on an evolutionary trajectory to get rid of them completely, which I'm totally in favor of. Of course, people with wisdom teeth would have to die and not breed in order to not pass on that gene, and I failed at that, sorry. And actually, one more, to end things on a slightly creepier note. You know this membrane that kind of just hangs out in the corner of your eye? Well, that is probably a vestige of something called a nictating membrane, which birds still have to this day, and is in fact a third and fourth eyelid. Thanks for watching SciShow. If you haven't, go to youtube.com/SciShow and subscribe, because we're very interesting here. If you'd like to ask us questions or suggest video topics, we're on Facebook and Twitter, and always in the YouTube comments below. Goodbye.
All right, so that's a little bit about vestigial structures. Actually pretty cool when we look at the importance of vestigial structures. These are structures really that aren't that important anymore. They are left over structures from a bygone era. That's what vestige means. So moving along to our next example of evidence, I'm trying to go through this a little bit quickly because we've got lots of videos to watch. We're going to look at what are called homologous structures. Homologous, you should remember homologous chromosomes are chromosomes that are the same size and shape and have similar genes, right? Homologous structures are similar structures that are used for different functions in different organisms. And homologous structures generally are seen as being developed from the same ancestral body parts. This results from divergent evolution. So if you think about having a common ancestor which is diverging in different in environments or different habitats, there may be a certain advantage to having a body part being able to be good at, for instance, digging, or another one being able to be good at providing speed during running, or another one that can provide lift for flight. So what we see is similar structures, different functions developed from the same ancestral body part. So let's take a look at a video here. This is one of the last ones that we'll see. This is about whale evolution. We're going to see whales actually have structures that are homologous to structures that are in our bodies. We're going to take a look at that as well. Get this video to pop up here. Let's see what we got. By the way, the questions that you want to be paying attention to, we should go back to that. We want to look at question number, or letter B, number 6, and then also the C, D, and E, and finally F that would be on page 94 and on page 95. And it looks like here's the video. I'm going to drag it down so it's a little bit bigger for you to see. And here we go.
Mammals first appeared on Earth around 200 million years ago, on land. Mammals are warm-blooded. They give birth to living young, and they breathe air. These are all adaptations to living on land. But whales and dolphins are mammals too. When Phil Gingrich began his career 30 years ago, he knew nothing about whales, and that was just fine with him. Gingrich's early interest in primitive land mammals eventually took him to Pakistan. It was there that he made the kind of find most paleontologists only dream about.
This is the original specimen. It's the one we found in about 1978. There are several things that strike you. One is it's very similar in size and shape to the back of a skull of a wolf.
But there was something odd about this skull.
If this wasn't here, I would think that this was an archaic carnivorous mammal, what we call a creodont. But it is here.
It was part of the animal's inner ear, and it had a distinctive shape. A shape found today in only one kind of animal: whales. The Sahara Desert is one of the driest places on Earth. But 40 million years ago, things here were quite different.
This used to be the sea. Just think of this being the current Mediterranean coast of Egypt, backed up about 40 million years, about 100 kilometers to the south of where it is today. Here's a Basilosaurus.
A 40-million-year-old creature already known to science. Basilosaurus lived full time on the water. After only a few days of digging, he made his second amazing find. It turned out that Basilosaurus had something modern whales have long since lost. The bones were small but unmistakable. A pelvis, a kneecap, even toes. This whale had a complete set of leg bones. It was dramatic evidence that whales had once been four-legged animals. Since Gingrich's finds, he and others have filled in more of this fantastic story. Scientists now think that the earliest ancestor of whales was similar to this 50-million-year-old wolf-like mammal called Synonyx. Synonyx was a predatory scavenger that lived and hunted along the shores of an ancient sea. Perhaps its descendants found the water a source of abundant food and a haven from competition. Over millions of years, front legs became fins. Rear legs disappeared. Bodies lost fur and took on their familiar streamlined shape. Since Gingrich's first find, named Pakicetus, the list of known transitional whales has grown. It now includes Ambulocetus, Rhodocetus, Durodon, as well as Basilosaurus. They reveal another element of whale evolution, the gradual migration of nostrils to the top of the head as whales adapted to breathing in the water. And bones aren't the only evidence of whale evolution. Their ancestry is also visible in the way they move. Fish swim by flexing their spines from side to side, like the shark. But mammals swim differently. This otter swims by undulating its spine up and down, in exactly the same way that whales do. And, as it turns out, in the same way that land mammals use their spines when running. Whales took with them into the water their ancestral way of moving, and we can still see it 50 million years later.
All right. So who would have guessed that a whale evolved from a land mammal that was somewhat like a wolf? Pretty insane. So again, big idea here, homologous structures are structures that are similar in actual shape and structure, but different in their function. These come from a common ancestor, and they result from divergent evolution. When you look at the bat wing and the mouse forelimb and the human arm, they are the same, bone for bone. All these bones are the same structures. They just are sort of morphed out in a slightly different proportion based on what the use would be. So those are homologous structures. The very last bit of information I need to share with you here are analogous structures. This relates to question letter F. That would be on page, I believe, 95 in your packet. So analogous structures are similar in purpose, but they're not inherited from a recent common ancestor, so they're not necessarily structurally identical. In this case, when you look at an example here, you have a butterfly that has wings and a bird that has wings. The structure is very different. The butterfly is made up of chitin. It does not have the same kind of skeleton that a bird does. It's got an exoskeleton. Bird's got an internal skeleton, calcified bone. So even though the wings share a purpose, they enable flight, they're not inherited from a recent common ancestor. The common ancestor of insects and birds is quite distant. In the case of analogous structures, the environment is selecting a particular trait. So it enables these organisms to be good at procuring food that is up in the sky, for instance. It opens up a new niche to them. This is an example of what's called convergent evolution. When you have two very different kinds of organisms living in a similar environment, they may evolve convergent or similar structures that are similar in their purpose. So, analogous structures. All right, I know this was long. This was the last screencast. You're not going to get this in chemistry next year, and we're letting you use your notes from this on your tests. So I hope you enjoyed it. It was hastily put together, not very well crafted, but there it is. So we'll see you in biology.